Okay, thank you for that nice introduction. I'm excited to be here. This is my first time at the IAD days. But as Rachel mentioned, I'm from the University of Pittsburgh, so not too far away. So it's great to be. Is it okay? So it's great to be here. Is the mic working? Okay. 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 Don't want it to fall down. Okay. It's okay. Can everyone hear me? Okay, in the back. Okay, great. So, so Rachel mentioned a little bit about my research. So my research broadly has focused on understanding emotional processes from a multi-method approach. My earlier work focused primarily on affective disorders like depression, anxiety, and more recently I've been branching out transdiagnostically into other populations, including clinical risk for psychosis and most recently autism. So today, I was trying to decide what I wanted to focus on today. That's why my title is kind of long and broad. But I'm going to focus on my most recent work in autism for the talk today. And I also am a clinical psychologist, as she mentioned, so I also do clinical work. So about 1 day a week, I also see patients for psychotherapy and assessment, and I also do training in cognitive behavioral therapy. So my research and clinical work often inform each other. Let's see if this still works. So just a little background about autism, because I'm assuming the majority of the audience may not know the clinical criteria for autism, but may probably have heard of it and knowing people in the population who have autism. So autism, according to the DSM, which is our Diagnostic and Statistical Manual, autism involves persistent deficits in social communication and social interaction, things like social reciprocity, verbal communication, as well as nonverbal communication. It often involves restricted or repetitive patterns of behavior, such as stimming, which is often thought to be a way to regulate oneself in overstimulating environments, as well as more restricted interests or activities. It's a developmental disorder, so symptoms must be present early in development, but it typically will continue through adulthood, but may manifest in different ways across development. Sometimes people are not diagnosed until later in development, especially for those who are higher functioning, when social demands might exceed their capacity, or they might also have learned to mask using different strategies later in life, especially for women. They might tend to use various social ways to mask their behaviors to sort of hide that they have autism. So often it can be underdiagnosed, especially for women with autism in adulthood. And symptoms cause clinically significant impairment in functioning, especially social functioning. And it can't be better explained by other intellectual disabilities or developmental delays, but it can include intellectual impairment, but not always. So about 2 to 3% of adults are, according to epidemiological research, have been diagnosed with autism, although those rates seem to be increasing. And we don't know all the reasons for that, but it seems to be a large part because there's an increasing awareness of autism and more people are seeking assessments for suicide. But we know very little about suicidality among individuals with autism. So, but we know that this is an important problem in this population, and there's major gaps in research, especially with adults. The majority of research has been in developmental samples, so kids. We know very little about how autism and comorbid psychopathology and suicidality manifests in adulthood. And prior research is pretty cursory. It's focused on really small samples of convenience or correlations with self-report measures, primarily cross-sectional studies. So there's been very limited larger scale studies, longitudinal studies with adults. There also has not been any studies with adults looking at neurobiological or physiological mechanisms that might contribute to emotion dysregulation or suicidality in daily life among adults. And then there's also this cliff in terms of the research in adulthood. So there's not as much funding for research on autism in adulthood, and there's been a lot less work in it. So even though more adults than children are diagnosed as autistic, only 3% of autism research is focused on adults. And many more and more teens every year with autism enter adulthood, and there's going to be upwards of 500,000 more adults with autism each decade if the rates continue to go up. But we have all these people that we don't really know much about how emotion is dysregulated and what are the risk factors for suicide, or also how can we treat them. So I also work in a clinical setting, and adults with autism often really struggle to find the right clinical services for them because many clinicians might not be comfortable working with autistic adults because they feel like they don't have the expertise, but most of the autism clinics are focused on kids, or if they have other comorbid psychopathology like psychosis, and autism clinics might not feel comfortable working with them either, so we really struggle to find clinics that really can work with this unique population. So I'm part of this Autism Center grant and ACE We call it ACE, the Autism Center for Excellence. The director at Pitt is Carla Mazevsky. So there's a few different projects. I'm going to be focusing today on Project 2, which I lead at Pitt with Lori Scott. And then there's a couple other projects. So Project 1 is going to be focused on developing a better self-report measure of suicidal risk in autistic adults. And Project 3 is going to be focused on neural circuits of emotional reactivity. And then we also have other clinical assessment core, administrative core, and our dissemination and partnership core. And that's a really cool part of this project because we actually interact a lot with the autistic community and our autistic partners. They actually reviewed a lot of our measures and gave us feedback on them before we started using them in our study. So it's been really great to work with the community to make sure that we're using measures that's sensitive to this population, that they are comfortable with, and to use their input. So in terms of the Project 2 that I'm leading at Pitt with Lori Scott, the focus is understanding how my emotion dysregulation manifests in daily life among autistic adults. So as I mentioned, most research in adults is primarily focused on retrospective questionnaires, but these are, of course, limited because they don't have as much ecological validity to capture daily stressors in the context of daily life. They also don't account for the difficulty that autistic individuals often have in identifying and describing their emotions. So there's also very limited research using psychophysiological methods in autistic adults and none in daily life that I'm aware of. So this will be the first study to look at that. So the prior work has only been in the lab with really tiny samples. So overall, we have a really limited understanding of how emotions unfold in daily life in autistic adults and how the corresponding physiological processes and how these things can predict proximal risks for suicide or self-harm or aggressive behaviors. And so in terms of the overall aims of the study, which is currently ongoing, so I won't have the answer to all these questions here, but I'll talk about some preliminary findings. So our overall aims are to characterize the different contextual features and components of emotion dysregulation in daily life across multiple response systems of self-report, behavior, physiology, and examine how components of emotion dysregulation predict harmful outcomes like suicidality, aggression, self-harm. And then our third aim is a center-wide integration across the different projects to develop personalized models of dynamic patterns of dysregulation in daily life integrated with data at other levels. So our target sample, data's collection is ongoing right now, but our target sample will be 300 participants from the community. We have about 50 to 60 so far. We're recruiting a wide age range from 18 to 65. 200 will be autistic or have like an autistic spectrum disorders broadly defined. So we'll include a wide range of severity. They have to be able to at least understand the study procedures and communicate verbally. But we were pretty liberal with our inclusion criteria. So some of them, do have some mild intellectual disability. Some of them do have some challenges communicating verbally. So some of them prefer to communicate with us over text or in written form. So we're able to accommodate that. And we also have 100 non-autistic, but they're not completely healthy controls because they do have both of the groups are able to have other mental health diagnoses as well, like depression and anxiety. And both groups are oversampled for recent suicidality. So at least 50 in each group will have reported recent suicidal ideation or behavior. So our protocol is a 14-day ecological momentary assessment protocol. I don't know how much you guys are familiar with EMA, but basically it's also called experience sampling method. Sometimes that term is used interchangeably, but basically use the device like a smartphone, like on the picture here, to give surveys to individuals throughout their day. So in this particular study, we have up to 4 physio-triggered, which I'll talk about more about that in a minute, and 4 random prompts when they don't have a physio trigger, so kind of randomly spread over the day. And we also have continuous passive sensing of electrocardiogram or ECG data, as well as electrodermal activity or EDA. I'm going to talk more about that. So the platform we use is MovieSense platform. It's a German company, and they provide integration with the sensors that they develop. So this is the ECG sensor here, and their own app, the MovieSense XS app. It interfaces with each other via Bluetooth so that we're able to do physiological triggered prompts. We're able to download the full raw signal. So for those of you familiar, this is like a full electrocardiogram sampled at 1,000 hertz. So really high quality data. And at each survey, participants complete a number of self-ratings. Most of these are rated on a 0 to 10 scale. So they rate their most recent positive and negative emotion, their biggest emotion in the past hour, as well as their self-reported level of arousal. Oh, sorry, that's the current one. They rate their current positive and negative emotion and arousal, and then they rate their strongest or most intense in the past hour, as well as associated contextual variables, including if they experienced any overwhelming sensory experiences or social interactions. And those are more like checkbox responses for the categorical ones. Then they rate, and if they, also the 0 to 10 scale, if they used any specific emotion regulation responses or strategies. They rate whether or not they engaged in any stimming or masking behavior, as well as any dysregulated behaviors like suicidal ideation behavior, self-harm, or aggression. And we do have safety protocols in place. So if someone identifies intent to harm themselves, it triggers an alert. And we have someone on call each week who gets like an alert on their phone. So they're able to call the person and do a safety assessment to make sure the person is safe. So right now, because we only have 50 people, I won't be talking about the suicide prediction just yet, but I'll be talking about some of the preliminary analysis with the other variables. So, and this is what our sensors look like. So we have the chest sensor for the electrocardiogram, and then we're also getting electrodermal activity, which we get from the foot. It's attached by the ankle, and then the sensors are on the foot. And we created this infographic for participants to show them so they have like preparation to understand what they're going to be doing, how they're going to be wearing the sensors so that they feel really comfortable and know what they're getting into. So we do give them a couple options for the chest band. So they can either wear, it's like a Velcro, like strap with dry sensors. It's kind of like an athletic, it's just like an athletic strap that athletes sometimes use. And then on the right is a different option. It's the same sensor, but it's plugged into these sticky disposable electrodes. And this is important for this population because they often have sensory sensitivities as well. So we found it really helped with compliance to give them choices if they wanted to use the chest band versus the electrodes. And some of them actually wanted to flip between the different ones since they were doing it for 14 days. It helped them feel more comfortable if they can like sometimes do the band, sometimes do the sticky electrodes, and the signals were pretty comparable. So in terms of the physiological signals we're collecting, so just to briefly describe these, the two main ones that we're interested in are respiratory sinus arrhythmia, which we're able to extract from the electrocardiogram. So respiratory sinus arrhythmia can be indexed by high frequency heart rate variability or RMSSD of adjacent R peaks. So this reflects parasympathetic activity. It's an index of vaguely mediated parasympathetic activity that's linked to the respiratory cycle. So as we breathe in, our heart rate speeds up, and as we exhale, it slows down, and this difference is reflected in this high frequency heart rate variability. And a lot of research has shown that this could be a potential transdiagnostic marker of self-regulation, so having more of it is better. Whereas electrodermal activity, this is also known as skin conductance or galvanic skin response. This reflects activity of the sweat glands, which reflects sympathetic arousal. It's often used in studies of stress, but it is not valence specific. So it can be any kind of arousal. It can be like really excited, positive affect, or negative. So one challenge with these measures, there aren't necessarily like emotion specific. So we also had the physio-triggered algorithms I mentioned earlier. So we worked together with the vendor MovieSense to kind of develop these for our studies. So we included one both for the RMSSD, so the heart rate variability measure, and then for EDA. So if they had a decrease in RMSSD or an increase in EDA, the person could get a trigger to give them a report at that time, with the idea being we can oversample periods of time when maybe they're feeling dysregulated. So we adapted the individual thresholds for each person and day because the original thresholds available by the software could only do really crude parameters such as, okay, there's a 20% change, then we'll do a trigger. But we found it didn't really work so well to use the same percentage across all individuals because then some people would get a ton of reports and some people would get none. So by using this adaptive algorithm, we were able to get a roughly even number per day per individual. So basically the day is divided into these sub windows too because we don't want any triggers to be too close together. So we had various rules to make sure, they were at least like 90 minutes apart and things like that. So we get like good coverage of the day and not annoy the person too much by having a bunch of reports in a row. So it continues to adapt. And we also have like a 20-minute delay in there. So we're not interrupting something during the most critical part of whatever's going on. So if they do get a trigger, it waits 20 minutes before it then prompts them with the survey to allow whatever's happening to kind of reach some sort of like conclusion. Although of course things might take more time than that. So this is just like a graphic of what that looks like. We also use different thresholds for high and low activity. It's something we're still thinking about the best way to incorporate physical activity, since if you're exercising, your sympathetic activity is, of course, going to increase. So we did have different trigger points for the high and low physical activity, and so which seemed to work well. And you can see here how the different triggers, how it's divided into these different parts of the day, so that we can get a roughly balanced number of triggers within each block of the day. And then just an example of when I was initially piloting this to see like kind of what sorts of examples we're getting at. So I initially piloted this with some students and have them write down like what exactly happened when that they were getting triggered. And it does seem like it's picking up on things that are like really stressful, like falling down in the snow, having a stressful conversation with a stranger or a roommate, talking to an ex, or being late for an exam and running to the bus, you know. So it seemed like it is picking up on things that are meaningful. So for the study so far, we have about 55 people in the data. I'll present here today, 47 autistic and 7 controls. So at this point, I wasn't able to do comparisons with our autistic and control group. So the analyses I'll present today are just within autism. We found I was actually surprised with how well our compliance and validity rates were, just given that there can be challenges working with this population. They have a lot of sensory sensitivities. So, but they actually did remarkably well, comparable to the rates that we have in the field study, which Kristen will talk about in later this afternoon. But we found that they were their compliance rates for filling out the surveys was over 80%, and their sensor wear time also over 80%. And in terms of the data validity, also close to 80% for both of the sensors. I also found in working with this population, they really cared about the study, I think because they know there's not much research with autism in adults, and they really cared about doing a good job. So we also had to be really careful too that we didn't that we were mindful of that, so that they didn't feel like compelled to do something that they weren't comfortable with, or that they were becoming overly anxious about doing a good job. So we also had to balance that, because they seemed to care a whole lot and they would get really upset if they weren't doing it perfectly. And there weren't any difference, at least initially, on compliance between autistic and controls, but again, we only have 7 controls now, but it is comparable to our other work. And in the debriefing results, responses were pretty neutral. Everybody reports a little discomfort wearing the sensors, but it wasn't anything like concerning. So today I'm going to talk about some preliminary findings we have, where I'm examining physiological associations with self-reported sensory experiences, stimming behavior, and masking in daily life in the sample so far, with a focus on the HRV data. So we know that sensitivity to sensory stimulation, stimming, and masking behaviors are common among autistic adults and kids. Autistic adults often report that feeling overstimulated, having a lot of sensory stimulation, can lead to feeling dysregulated. And stimming behavior, they often report as self-regulating. So it's something they might do in particular when they're feeling overstimulated and stressed. While masking of stimming, so hiding it from others because you're embarrassed or don't want others to know, that can often be experienced as depleting, and we're depleting your emotion regulation capacity or feeling drained. So this HF-HRV or RSA may be a useful marker of depletion of this self-regulation capacity associated with sensory overstimulation and masking, and it might be associated with increases in regulation if stimming does have this purported regulatory function. But no one has yet examined this in adults with autism, and no studies have looked at this in daily life. So I predicted that lower high-frequency heart rate variability reflecting lower parasympathetic activity would be associated with a report of recent overwhelming sensory experiences, and that masking behaviors would be associated with subsequent depleted HF-HRV at the next time point, and that stimming would be associated with subsequent increases in HF-HRV. So we use mixed effects modeling or multi-level modeling with 50 autistic adults. About most of them are female at this point, which is interesting, actually, because most people with autism are male, but I think we had a lot of females in this study given our focus on suicidality. So we assessed within individual associations between sensory experiences, masking, and stimming, using the 60-minute period prior to the survey and then the 60-minute subsequent to the survey. So I found that the past 60 minutes, if they reported experiencing overwhelming sensory experiences, that predicted that they would also engage in stimming in the same period. So that's consistent with what we thought. We also found that overwhelming sensory experiences did not predict subsequent high-frequency heart rate variability, contrary to expectations. And the stimming did not predict increases in HF-HRV. So people are doing it when they're feeling overwhelmed, but it's not really having an impact on their physio, at least not the time windows I was focusing on initially. And then lower prior high-frequency heart rate variability in the 60 minutes prior to the prompt predicted higher recent stimming but not masking behavior. And then, but masking behavior in the social context did predict lower subsequent HF-HRV, suggesting that masking is depleting this regulatory response. So some support for the hypotheses. And again, this is only like 50 people so far, so we may be finding more effects when we have our full sample. So this is, and I didn't do the EDA yet. I started looking at the electrodromal activity, but so far the effects weren't coming out significant. But I hope to continue those analysis once we have the full sample to see if there's any differences in the sympathetic activity as well. So this is the first study to examine ambulatory physio in daily life with autistic adults. We found that stimming did not predict increases in HRV, even though, but although it did, lower HF-HRV predicted increased stimming behavior. So it seems that when people are more depleted, they're using stimming more often, but stimming does not necessarily seem to increase HF-HRV, at least not in the very next time window. Maybe it would if we look further out. Masking behavior did predict lower HRV, which is consistent with autistic individuals reporting masking to be depleting. So that seems to be like an important clinical finding. This seems to have important implications for understanding physiological processes underlying sensory experiences and behavior in autistic adults. And then later on, we can link this to some of those more problematic and concerning risk behaviors like suicide and self-harm. So I'm also planning to do other lagged analysis. So here I was just focusing on those immediate time windows before and after, but things might be happening at other time scales. So I am planning to look at other, time points before and after. And also when we have more controls to compare with the non-autistic group, as well as the influence of other mental health conditions as moderators. And then when we have more data, we hope to examine the proximal risk for suicidal behavior, self-harm, and aggression. And there's a lot of data here, so there's a lot of potential applications for using things like machine learning to combine variables in innovative ways to enhance prediction of our models by combining the self-report behavior, like we have accelerometry, physical activity, and the physiological data to help enhance prediction. So that's it for the autism part of the study. So just to kind of briefly mention some of my other collaborations using ambulatory psychophysiology. So Kristen Gandy is going to talk about the field study in the next talk. So we used a lot of similar methods for that one in distress disorders. And I also work with Leslie Horton at Pitt using some similar methods to understand stress dysregulation in adolescents at elevated clinical high risk for psychosis. And then with Stephanie Stubb and Lori Scott at Pitt, we're also doing a dyadic study with couples where both of them wear the sensor. And if one partner is triggered, it'll trigger the other one too. So they're doing like a survey at the same time. So it's sort of like a yoked dyadic study. It's really complex. I was surprised we were able to get it to work. It's pretty cool. And then we're also getting like audio recordings with the ear of those like conversations like during conflict situations. So that'll be really interesting because we'll be able to link the physiology between the two members of the couple. And then another work that's kind of unrelated, but just to give you guys the highlight of some of the other work I'm doing that's more clinically applied, where I'm also collaborating with people in affective computing at Carnegie Mellon University. We recently, in the past couple of years, finished an NSF-funded clinical trial comparing cognitive behavioral therapy and interpersonal brief psychotherapy for depression. And then with our collaborators at CMU, we've been using affective computing, using automatic like face posture and vocal characteristic coding between the patient and the therapist to better understand synchrony and other measures, nonverbal dynamic measures in relation to how that affects treatment process, like working alliance, as well as outcome. So we have some initial papers that have come out with one of the students here. If anyone is interested in some of my other work, that might align with some of the affective computing stuff that may be of interest here. And that one, this is also like in a clinical study, so I am doing a bit more applied work as well. And just to get some acknowledgements for my funding and list here specifically my collaborators in the autism work and the React lab that we collaborate there for the autism study. And yeah, that's it. Thanks, everyone. I realized I couldn't see the clock here, so hopefully I'm on time. Is it not? Oh, it's for the questions, yeah.
So maybe I'll have a few. Okay, we can open the floor up for questions now. Just raise your hand and we'll bring them up.
There's one in the middle there. Yeah. Thank you. Yeah, that's a great question, actually, because that. Oh wait, are they gonna be able to hear me now? Yeah, that's a great question, actually, because that is something we struggled with initially. So we usually, and Kristen will talk about more our design in the other study, but we usually use several positive and negative affect terms in order to assess positive and negative emotions. So specific discrete emotions like sad, happy, scared, where people rate that on like a Likert or like a 10-point scale. But we found initially that our autistic participants really struggled with that. They actually found it really stressful and really aversive because they had trouble distinguishing between all those different emotions. So then we decided to just really simplify it. So now we're just using, I don't know if anyone's familiar with the SAM ratings, but basically there's like a little picture of this little man and it goes from negative to positive, like a frowny face and a smiley face. And then we use it for arousal too, to show really calm and really kind of like aroused. And so that worked much better for our population so that they felt much more comfortable being able to do more of a just a bad versus good. Of course, we lose some nuance in our ability to look at specific emotions, but we felt it more important, like we didn't want to stress them out, with trying to get at all these individual emotion words when they really didn't feel like they could do it. So that was an example of one way that we accommodated that. We also we showed that our autistic partners all the language, the questions and the instructions, and we got a lot of feedback where some of the wording might have been ambiguous or confusing. So just being really like straightforward and direct with the wording and just not allowing like anything that's kind of confusing or ambiguous. So we used a lot of that feedback to try to just make it as clear as possible. So does that answer your question? Yeah, thank you. Mm-hmm.
So I was interested in your oversampling of individuals with
Mm-hmm.
societal sponsor
Yeah.
ideation. My question is, what do you hope to do with that data?
Mm-hmm.
Thinking from the point of view of intervention, and what kind of models are you thinking about
Yeah.
using?
Yeah, good question. So this is not an intervention study, but of course, if someone reports in the context of the study that they're going to hurt themselves, we will intervene if we need to. But the hope is that we'll get enough. We oversampled for it, so we would get enough reports within the momentary data of people feeling suicidal or feeling some intent to die or feeling like an urge to hurt themselves. So we wanted to get enough data so we can look at proximal risk factors for increases in those behaviors that we could potentially use in an intervention later on to identify these risk factors, whether it's physiology or something else, before it escalates into suicidal behavior. So that would be the eventual hope of what we can do with this data. So the initial analyses we propose are multi-level as well as some gimme analysis, but I think there's a lot of applications for machine learning or other types of advanced analytic approaches. So I'm also open to other ideas. I'm sure there's a lot of ways that we could look at this data.
So how are you trying to get the data like this? I saw some sensors over there, like
Mhm.
was trying to think of chest. So how are you reading that? And yeah, like do you read the blood pressure or like how do you
Mhm.
convert that physical to
Mhm.
something into mathematical thing?
Okay.
What do you think?
Yes, good question. We are able to get some snapshots of the data through Bluetooth. The Bluetooth connects to the phone and that uploads a snapshot to the cloud. So we're able to at least see if people are wearing it, if the data is valid. But because the data is so huge, we get about 5 gigs of data per person. We have to wait till they come back to the lab and it's through a USB direct connection to the computer that we download it, the full raw data, just because it's so large and it's sampled at 1,000 hertz, where we get the electrocardiogram, the electrodermal, accelerometry, all these different signals. So it's a massive amount of data. So we do have to download it manually at that point. But it is organized into, like comma delimited time series data. So you can use it in any kind of software program. It's very flexible. And For this, we've been binning it like in 1-minute bins for our analysis, but we also have like the second by second data, like at 1,000 Hertz sampled rate. So you can look at it in many different ways. It's very flexible. But you asked about blood pressure too. We don't have blood pressure because that's a different type of sensor, but we don't have that. Let's make sure I don't fall off the stage here.
I have a question about that as well. So it seems they had one vendor that is, I saw, I logged into their website to look that they had one waveform. But are you looking at other like watches and devices because in order to get a large population to understand like
Yeah.
so what's the kind of the future
Yeah.
yeah so you be able to convince
Yeah.
behaviors or against work with Apple Watch to say we have this
If they give me the raw data, they won't give it. Yeah, it's an important question. The problem with a lot of these consumer grade devices like Fitbit or Apple Watches, they refuse to provide the raw data to researchers. So they do some pre-processing on the device and do some proprietary algorithms. So this is a problem for research for being transparent and reproducible if we're not if we're not able to see the raw data and apply our own processing to it and to validate what is done. There's also issues with any of the wrist-based devices or like the R ring. It's not a full electrocardiogram. It uses pulse plasmography, like a PPG signal. So it's a little bit harder to determine what is artifact or what is just like a misbeat or what is actual heart rate variability when you can't see the full electrocardiogram. And there's a lot of quality differences with the devices. Like one that's really popular is Empatica, but we recently published a paper led by my postdoc here where we showed that the valid data rate of Empatica compared to MovieSense is quite low in daily life and really unacceptable, especially for HRV. And the sampling rate is quite low. So a lot of these devices, we really have to be careful, even if they say that they're marketing it for research. We have to really look at the quality of that data and what we're getting. So it's really important to do those kind of tests. But I really hope that those other devices will continue to be developed and make the raw data available to consumers because I agree this type of sensor is more burdensome than something like a watch. So I hope to see more development. But as of now, those devices just are not as good for those, various reasons. And you can look up our recent paper where we did some really detailed comparisons. But good question.